Hey, what's up, class? Way to get through test numero dos. Grading it looks good. I think a lot of you guys studied and a lot of you guys got that the material and the concepts in our second unit. So, very proud of you guys. Way to go. So, we finished looking at how cells use energy, how they make ATP out of food, and how plants can make their own food, sugar, out of sunlight using energy from sunlight. And now we're going to be moving into a unit that is going to be looking at cell division, how the cells in our body divide, how we pass on genes to the next generation of offspring, and exactly how that works. So we'll be looking at 3 chapters in this unit, chapters 6, 7, and 9. We're going to skip 8 just for time's sake, and we honestly might end up skipping chapter 9. I would rather spend more time with you guys on the evolution part of the course than on other stuff. So, yeah, let's get into chapter 6 and cell division. So first, a brief overview of chapter 6. Has 5-ish different sections. The first one is what is cancer. Second is how cells pass genes and chromosomes to daughter cells after they have divided. So in here we're going to look at why do cells divide and what happens to DNA when cells divide. So we're going to look at these 2 sections on this online lecture, and then on Tuesday we'll pick up with section 6.3, learning about the cell cycle and mitosis and how cells actually go about dividing. We'll get into the control of the cell cycle and how cells control cell division. And then finally we'll look at meiosis, which is a special type of cell division. And we'll see how cells pass on genes, or how we as sexual beings pass on our genes to the next generation. Okay, so let's begin with cancer. What is cancer? So I think this is really interesting because at first cancer might seem like an infectious disease, something you get from the external environment, right? Like a viral, bacterial kind of deal. But really, all cancer is cells in your body that are dividing in an uncontrolled manner and results in a tumor that can then spread. And this happens because your cells accumulate mutations over time, and eventually those mutations can affect genes that control cell division. And so these cells grow out of whack, and they can just keep growing and eventually take over vital organs and kill you. And believe it or not, cancers occur in most long-lived organisms. So here you see dogs, boxers, for example, are particularly prone to tumors and cancers. Plants and trees can get tumors. And mice, they normally don't live long enough to get tumors, but in the lab, we can actually induce tumors in mice. And we actually use mice to study cancer biology and how cancers grow and how we can treat them. So yeah, basically if you live long enough, you'll get cancer, is the whole point, and we'll learn why that is. Okay, so like I said, cancers arise because of mutations in genes that control cell division. So these mutations can lead to hyperactivation of genes that stimulate cell division, which is like the gas, pressing on the gas of the system. Imagine the cell is like a car, and so if you press the gas, that means you're stimulating the cell to divide. So if you hyperactivate these genes, it's like you're stepping on the gas full force, but you still have brakes in the system. So mutations will also inactivate genes that stop cell division, that are there whenever the cell senses that it's dividing too quickly or that it's not ready to divide. These genes will stop cell division. And so what happens as mutations occur in these genes is you hyperactivate the ones that simulate cell division, so you're pressing on the gas, and eventually you cut the brake lines. You kill the brakes. So now it's like that movie Speed with the bus that is going full speed and can't stop, right? So, and just so you know, mutations are changes in DNA sequence. So any change in the DNA sequence of a gene, and a gene is one chunk of DNA that has the information to make one protein. So if you change one of the letters in the DNA sequence, you'll eventually change the protein, right? And that might lead to hyperactivation of the proteins or inactivation of the proteins that control cell division. And these mutations can be errors that are made during DNA replication before the cell divides. So these can happen naturally. That's why if you live long enough, even if you live in a bubble and you don't touch the external environment, you will get cancer, simply because every time your cells divide, they copy the DNA, and they don't do it perfectly, so mistakes happen, mutations happen, and eventually some of them will lead to cancer. Or mutations can come from contact with an agent or chemical that causes mutations called carcinogens, and we'll touch on this at the end of our lecture today. So when genes that control the cell cycle are mutated, when the DNA sequence is changed and that sequence changes the proteins that are encoded by the gene, the cell can begin to divide in an uncontrolled manner and form a tumor. So here in the figure you have normal cell division, where one cell becomes two cells and those cells divide. But eventually you can accumulate enough mutations that you become cancerous. So this cell is going to eventually divide out of control and form a tumor. And tumors are just masses of cells. So you can have a tumor that's benign, where it's not affecting surrounding tissue, and if you find a lump on your skin, you go to the doctor and it's like, oh, it's a tumor. It doesn't mean you're going to die. Could be a benign tumor that's not affecting the surrounding tissue or organs. And so these are non-cancerous tissues. But they can also be malignant if that tumor invades the surrounding tissue. So like if it begins to invade the actual, for example, muscles that the tumors might be growing next to, or they can begin to invade the lung. And eventually these malignant tumors can metastasize, which means that the cells break off and form new tumors. So eventually, as the tumor grows, remember, as cells divide, they accumulate mutations. So if tumors are cells dividing out of control, eventually they'll accumulate more mutations and it'll mess up these cells even more. And eventually the cells will acquire the ability to break away from the original tumor and start new tumors in other tissues or organs. And that's how you die. When you die from cancer, you die because tumors have formed in vital organs, such as in the brain or in the liver or in the lungs or other places. So for example, if you get breast cancer, you're not going to die because you have cancer in the breast. You die because that cancer will eventually spread to vital organs and eventually destroy those organs. So let's talk a little bit about risk factors of cancer, because it's important to know, all right, if it's such a deadly disease and it can happen to anyone, what are risk factors? What can increase my risk of cancer? So there are lifestyle risks. So eating a low fiber, high fat diet has been scientifically shown to increase your risk of cancer. It doesn't mean that it causes it, but there's a correlation. Like we discussed in the first chapter, correlation does not equal causation, but there's a link there somehow. So a high fat, low fiber diet can be seen if you eat fast food. This is you, right? And I every now and then I will definitely enjoy a what did I have the other day? Oh, a Chick-fil-A sandwich. And Chick-fil-A, believe it or not, even though it's chicken, it's still fast food, all right? So try to limit your fast food, not because it's just it can cause you to gain weight, but because it's been shown to be scientifically linked to heart disease and cancer risk. Also, lack of exercise increases your risk of cancer. Exercise is beneficial for the body in innumerable ways. So let's not be sedentary people. Let's be people who walk around and who are active and enjoy the outdoors and keep our bodies healthy. Um, your risk of getting cancer also increases with exposure to carcinogens such as smoke and cigarettes, and really any kind of smoke. So people that say, "Hey, smoking weed is less harmful than smoking cigarettes because they're less carcinogens," that's a lie. There's still carcinogens in marijuana smoke. Not as many actually as cigarettes. It is not as bad for you in that regard, but it still has carcinogens, same as a hookah. So if you smoke hookah, you're putting carcinogens in your body. Um, Exposure to UV rays is also shown to cause skin cancer. So this isn't just sun rays. When you go to a tanning salon, you're sitting under UV rays that are literally mutating the DNA in your skin cells, and so you are guaranteed to get skin cancer if you are a sunbather, consistently sunbathing without protection, right? So you should, and protection is not tanning oil. Tanning oil actually enhances the effects of the sun. Protection is sunscreen. Love this little graphic of a tanning bed. So true. Cancers can also, you can be at risk of cancer if you inherit a mutation that has been shown to cause cancer. So for example, Angelina Jolie is mentioned in your textbook. She has a mutation she inherited from her mother that puts her at risk of getting breast cancer. And so she actually had a double mastectomy where she removed both breasts so that she would decrease her risk of cancer, because she had the gene that gave her an 87% chance of getting cancer. High chance, right? Don't want to risk that. And there's other mutations you can inherit. So if you have a family history of cancer, you should be aware that it is a possibility that you have inherited that risk. And then finally, aging. If none of these things get you, you're still not safe, because ultimately aging, if you live long enough, you will get cancer, because you will eventually, as your cells have divided for thousands, if not millions of times, by the time you're an old man, they will have acquired by chance mutations in important genes that control the cell cycle and cell division. Something interesting too from your textbook is how risk factors can work together to enhance the activity of that carcinogen. So, for example, together, smoking, tobacco, and drinking alcohol have a greater risk than either one on its own. And this is from a study, right? So this is a synergistic effect where it's not just 1 + 1 = 2, it's 1 + 1 = 5. And you should be aware that if you are actually engaging in a lot of the lifestyles or exposing yourself to carcinogens that we just mentioned, they could be acting together to give even greater effects than they are on their own. All right, so cancer is a disease of uncontrolled cell division. So let's look at cell division, and first let's look at why cells divide. So cell division is responsible for really important life functions such as growth. So when you go from one cell in your mother's womb to 37 trillion cells, more or less, that has been actually estimated and published in a scientific paper. A guy who went out and actually calculating the density of cells in different organs, adding it all up together, and estimating that we have around 30, give or take, 37 trillion cells in our body. So going from 1 to 37 trillions, that is happening because of cell division. Replacing dying cells, so whenever you scrape yourself and have a wound, the wound is actually healed by cell division. New cells come in and replace the ones that are dying or have died. Same thing with people that drink alcohol, too much alcohol. That alcohol kills liver cells, and those liver cells are replaced by new ones. And eventually, because cells divide, if you're drinking constantly, cells divide so much in your liver that you will accumulate mutations and eventually get liver cancer. So that's why people that alcoholics get liver cancer. Cell division is important for regeneration, like regrowing limbs. So in this figure here, you have an example of a newt, and newts, salamanders, and other amphibians are known to regrow limbs if you cut them off, the whole thing with fingers, as if it was new. And scientists are investigating this, trying to find the genes responsible to see if humans have the same genes and these genes can behave in a similar manner, to where now, instead of having vets who have come back from war with missing limbs, we can actually take them to a hospital, engage these genes that are involved in this process, and help them regenerate lost limbs. And then finally, cell division is involved in reproduction, so both asexual and sexual. So asexually reproducing organisms, like the single-celled organism in this figure here, reproduce simply by dividing, and boom, now you have two single-celled organisms. Multicellular, sexually reproducing organisms like you and I, we actually, our sex cells, sperm and eggs, go through a cell division process. So cell division is crucial for sexual reproduction. Okay, so now that we know why it's important, what actually happens to DNA when cells divide? So DNA is crucial, right? Remember, DNA is essential for life. It contains all of the blueprints and all of the recipes for the proteins in the cell. So if you lose it, you can't make those proteins. And remember, proteins are the workers of the cell, so if you don't have the workers of the cell can't do its business and it dies. So the cell tries really hard to keep that DNA intact. So when you're dividing, when the cell divides, it has to make sure that DNA is preserved and that each cell resulting from the division has its own copy, right? So it has to make sure that the new daughter cell has its own copy of DNA. So before cells divide, they have to, one, copy their DNA, which is called DNA replication, and two, condense the DNA into chromosomes to help separate each copy of DNA. And this is called DNA condensation. So let's look at these two things and then we'll finish our lecture. So DNA is replicated because each cell needs its own copy, right? The way I put it here is the new daughter cell, because this one cell is essentially copying everything inside it and then dividing and giving rise to a new daughter cell. So the new daughter cell needs its identical copy. For this to happen, what the cell does is it unwinds DNA, and then it separates the strands of the double helix, and then each strand, so here in red, each of the original strands are used as a template to make new strands. So a template is like an original document that you can use to make more copies, right? So think of it as a stencil. You have a stencil that is a template for the actual letters that you're going to make. So, so what happens after the DNA is replicated, you have two identical daughter molecules, each one with one parental strand in red and one new strand in purple. This is called semi-conservative replication, because only one of the two strands in each of the new DNA molecules is from the original. So you conserve one copy. So that's why it's semi-conservative replication. Semi because you're only conserving one of the original strands in each of the new DNA molecules. So how does the cell actually make the new strands? The cell uses an enzyme called DNA polymerase to synthesize the new strand of DNA in each DNA molecule. So what this enzyme does is it moves along each strand of the unwound double helix, as you can see here, and add complementary nucleotides to each strand. So what do I mean by complementary? And by this point, I know I'm going fast, but I'm going fast because by now we should all be comfortable with a lot of this terminology. So what I mean when I say nucleotides, I'm talking about the monomer of DNA, right? And the monomer, these nucleotides are made up of a phosphate, a ribose sugar, and a nitrogenous base. And so these bases, you have 4 of them, so therefore you have 4 different letters in the genetic code. and you have ACTG. These bases are complementary, so A will always pair with T and C will always pair with G, always. All right, so here, this is from one of our earlier lectures, and you can see this is a double-stranded DNA molecule while it's still together, and you can say that A always pairs with T and C always pairs with G. And they interact together, these bases, these nitrogenous bases, interact together via hydrogen bonds, right? Okay, so basically, that's how DNA polymerase knows what nucleotide to add next. It simply uses the complementarity of these bases. So for example, the next nucleotide in the strand on the left would be whatever nucleotide or base is complementary to the one on that original strand. So that is how the original strands in red serve as a template for the new ones in purple. And then we'll watch this video on Tuesday, but I just wanted to show you the link. You can go to this. These slides are now on Blackboard, so you can copy the link and go to YouTube, watch the video, and see DNA replication in action. Okay, so we looked at replication. Now let's look at condensation. So normally in the cell, DNA exists as chromatin. Chromatin is uncondensed DNA wrapped around a few proteins, but it's like silly string or spaghetti. Just it's a mess. You can see in this picture, this is an electron microscope picture of chromatin in the nucleus, and it is a mess of a web of DNA. But as soon as cell division begins, chromatin fibers begin to coil and condense into chromosomes. So these are the characteristic X-shaped chromosomes that you hear about and that we talk about in biology. And you can see here, we'll actually talk about chromosome structure later on. So you'll know exactly what's going on. But just know that a chromosome is condensed chromatin, right? So this is a cool illustration from another textbook that I'll use. But this is chromatin. Here you see the naked DNA. It interacts with proteins called histones, so it's wrapped around these proteins, and it forms like beads on a string. And then these coil some more to form chromatin. So there's chromatin. That's how DNA normally exists in the cell. And then as soon as cell division begins, the chromatin fibers begin to coil and condense into chromosomes. So there you see on the bottom the X-shaped chromosome. And this is a replicated chromosome. We'll kind of break this down in class on Tuesday, but in our lab, the lab I teach, we talked about this, so hopefully you kind of know what's going on. Okay, and you can see this is a light microscope image of a cell, of two cells. The one on the left has not begun cell division, and so the chromatin is uncondensed. It is that mess of spaghetti just jumbled up in the nucleus. And then the cell on the right has begun cell division, and its DNA has already started to condense. It's already finished condensing into chromosomes. And now, okay, so the reason it does this is so that it can divide the DNA content neatly, basically. And we'll touch on this on Tuesday. But just know the reason that the cell condenses the DNA before cell division is so that it can divide it neatly. By this point, the DNA has already been replicated, and now you're going to divide it. And here's another video we'll watch on Tuesday, but I want you guys to go ahead, copy the link on the PowerPoint slide, put it on YouTube, and watch this animation of DNA condensation. Really cool. All right, so let's put it together, replication and condensation. So before the cell divides, the DNA is replicated. Basically, the cell gets ready for division by replicating the DNA. So you have your chromosome, you unwind it in order to copy it, and you end up with two identical copies of the DNA. And DNA exists in chunks called chromosomes. You have 46 of them in the cell, so you copy each one of these. each one. So now you end up with 2 identical copies of each of the 46 chromosomes. And you'll see that each identical copy is called a sister chromatid. And these sister chromatids, after they're copied, they're actually going to be united at the center at the centromere, which are those 2 balls that are touching each other there. And after DNA has successfully replicated, after the cell has successfully replicated the DNA, it's going to enter cell division. So as soon as cell division begins, the DNA is condensed into chromosomes. And you have here a replicated chromosome. So each of the sister chromatids are identical to one another because they were replicated, right? And as cell division continues, what it's going to do is it's going to separate these 2 identical chromosomes so that one of them goes to one cell and the other one goes to the other cell. So it's basically duplicating the information and then splitting it during cell division so that each cell can have its own copy of DNA. And obviously this is happening for each of the 46 chromosomes in the cell. So we'll talk about how cell division happens and how the cell actually does this on Tuesday. But I hope you enjoyed this one. Try to keep it short for you guys. Enjoy your weekend, and I'll see you guys on Tuesday.